So last up we have Andrew Harrington. Where's Andrew?
Okay, so the lesser horseshoe bat, so called for the shape of its nose, is the rarest of the 9 bat species present in Ireland, limited to the counties of the west coast. It was once thought to be one of the commonest bat species in mainland Europe, but since the 1950s it declined massively due to pesticide use and habitat destruction. And it's actually thought that the Irish population, estimated to be 14,000, is actually one of the biggest and most important in Europe now today. So the way that the Irish population is monitored or counted is that bats are counted in the summer colonies where the females raise their young. Now, by itself, that data isn't enough to actually arrive to an estimate. So what we have to do is there are a few assumptions that we take. So one of the important ones is that it's assumed that all of the females in the population are present in these colonies, whereas most of the males, or a large proportion of them, aren't actually counted because they're present in smaller scattered sites. So it's thought that in these sites that are monitored, actually only 25% of the bats are male, which translates about a third of the entire male population actually being counted. So the problem with this assumption is that it's actually based on very little actual information. It's basically a guess. So this is the question I was interested in looking at, especially as DNA technology is very commonly used now in wildlife studies, and bat droppings, which are very easily collected in these colonies, are a very good source of DNA. This is the technology I wanted to apply to answering this question. So what I did was I visited in summer 2016 6 lesser horseshoe bat colonies in Ireland, and I gathered 1,300 droppings. So the number of bats in total was about 500 roughly. I designed DNA tests for this bat species, so to identify the bat species, to make sure I was getting the right species, to identify males and females, and to identify individual bats. And then I applied those tests to the bat samples I collected. So I extracted DNA from all of these bat droppings, identified the species, identified all the individual bats in each colony, and sexed them. And when I had done that, then I was able to actually look at each colony and arrive at a proportion of males, or to find out the proportion of males present in each roost. So what I found was that the actual proportion of males present in these bat colonies varied a lot, from 9% males up to 72%, okay? And most interestingly, actually, was out of the 6, there was actually well over the actual assumed proportion of males present. And actually, on average, there's near to 50% males are being counted in these colonies. So what this implies is that our Irish population is actually a lot smaller, over a third smaller than it was, so nearer to 9,000, which is a serious concern for the conservation of the species, not just in Ireland but in Europe. But it does mean that at least in future we'll have more accurate population estimates for the species. Now, the one last thing that should be said is that probably a little bit more work needs to be done to get more data. Okay.
Thanks very much, Andrew. Could I ask, what would the challenges be in adapting this methodology for a different species?
Well, that's a very good question. I mean, it depends, because these would have, they would roost in different kinds of buildings, like these roost in kind of abandoned kind of farm buildings, let's say, where it's going to be very easy to just get in and collect the droppings, where some species will actually live in trees, let's say, or under, like, between the slates and felt of a building, let's say, so it's going to be an awful lot harder. So you're going to have to change how you collect your samples, so your bat dropping samples, and as well as that, you're going to have to design a different set of DNA tests as well. But that should be very doable. It would take more work to kind of do it for a different species. But it's, in theory, it's something that could be done for any bat species or any wild mammal species.
Okay, thank you very much, Andrew.